Hello students, once again a very warm welcome to Misostudy sessions. So in today's class, we will be discussing about certain questions from the chapter Fundamental Unit of Life. We all know what is fundamental unit of life, that is a cell. A basic unit of life, our whole body is made up of millions of cells. Cells, they combine together, they form tissues, tissues combine together, they form organ, and likewise there is a hierarchy. So organ, they combine together in system, and likewise all these organs, they combine together and they form as a system. Like we have as a digestive system, so many different organs, they are present. They work together, they do certain kind of a proper function, right? And let's take an example of a digestive system only. They help in digestion. Though many organs are involved, some help in secretion of the bile, for example liver, some store it, whereas many other functions are done by the pancreas as well. So we have already discussed about the whole chapter, and we have also talked about the different type of cells, starting from a multicellular organism to a unicellular organism, to different type of cells, like earlier or some organisms they are having prokaryotic type of cells, to those organisms which are having a eukaryotic, a modern kind of cells. What are the different cell structures present in them, that all we have already completed. Like, I am hoping that you all must have seen the lectures which I have already delivered in the last few sessions, and now I will be focusing on the certain questions. 10 questions, each questions from the different categories, and I will tell you how to attempt such type of question. I will not go into detail. If I will go into detail, so that will take hours to tell you how to attempt such 10 questions, right? We will be focusing on the few of the important things. I will tell you how to write the things, and as you all must have seen the sessions, so hope you will be knowing the answer as well. So let's start the today's session with question number 1. So this is a question number 1. First of all, I have divided the whole 10 questions into different categories. First 3 questions are of 1 marks, then again 3 questions are of 2 marks, then the 2 questions from 3 marks and 2 questions from the 5 marks, right? This is how we will proceed. Let's focus on 1 mark question first. Question states, state 2 important functions of nucleus, right? You all know that nucleus, they act as a brain of a cell. All the activities which happens in our cells, whether it is related to cell division or it is related to any of the metabolic activity, everything is controlled by the nucleus. Even a nucleus has a DNA as well. Here the nucleus is written, that means I am talking about a eukaryotic cells. That means the DNA is present in the nucleus. So can I say that it acts as a hereditary material as well, or it is a storage of hereditary material? Yes, this is the right. So if such type of questions come, how you have to attempt? They are asking only 2 important functions. So you should write 2 important functions only, and try to write 2 important questions in a point-wise manner. Don't write stories. No one is interested in your stories. Just focus on the marks, like it is of 1 marks, so 1 or 2 lines, that is enough. No need to write a whole stories, right? So if such type of question come, what you have to write? For this, the answer is, state 2 important function of the nucleus. Nucleus controls all the metabolic activities of a cell. All the metabolic activity, whether it is related to digestion, respiration, excretion. Somewhere or other, you know that when a DNA is present, it leads to the formation of the protein. For that, we need that mRNA should be formed, and that mRNA is formed in the nucleus only, and later on it comes out and the translation happens. But who is the basic machinery? The basic machinery is a nucleus only, in that the DNA is a basic machinery. So it controls all the metabolic activities of a cell. So second important function, this is the first important function, this is the second important function, is that it controls the very important process which is a cell division. It controls a cell division. Now question arises, what is a cell division? One cell has a capacity to form the 2 cells. Meiosis, mitosis, everything is controlled by the nucleus only. Right? Third important process, because they are asking only 2, so this is just a class for you. I am not writing any question. So what you have to, because I'm writing the third one, this should be there in your mind, because I'm giving you 3 at least in the till the examination, till the exams will be there, you will at least remember 2 if such type of questions come. So the third answer in this case is that it acts as, or heredity material, because it has a storage of DNA present in it, right? So these are the 3 important functions of a nucleus. Hope this question is clear to you. If you know the answer, that is well and good. That means you will score at least 1 mark in your exam. So let's move on to the second question. Which organelle is called as a factory of ribosome? So hope you remember that session when I have discussed about the nucleus. So as you know, in the nucleus, one area is present, one darkened region is present, which is called as a nucleolus. Nucleolus. Right? Hope you remember the diagram of the nucleus. That nucleolus is present, and that nucleolus, in this, synthesis of ribosome occur. Synthesis of ribosome. That is why the correct answer you have is a nucleolus, right? Correct answer you have is a nucleolus. So nucleolus is considered as a factory of a ribosome, because in this the synthesis of all the majority of the ribosome occur. Hope you remember the diagram, just giving a rough diagram. Suppose this is a whole cell, right? In that a nucleus is present, on that a darkened structure which is present, that is called as a nucleus. Nucleolus. This is a nucleus, and this is a cell membrane. Right? So this is a question number, this is a second question, question number 2. This is how you have to attempt. Just a single word, that is a nucleolus, will give you a whole 1 mark. Let's move on to the next question. So this is question number 3, which is also of 3 marks. Sorry, 1 mark. So what is a plasmolysis? Plasmolysis. Listen very carefully. Plasmolysis is a process by which all the water which is present in the cell, that comes out. So in this case, what exactly the plasmolysis is? Suppose I have one beaker. Just have a look. I am explaining you what exactly this plasmolysis is. I have placed a cell in this. This is a plant cell, and plasmolysis always occur in case of the plant cell. Right? This is a plant cell. Right? In the plant cell, as you can see, this is a nucleus present, and we have already discussed that this is a cell wall present, and below the cell wall, this you can say protoplast. If you say this is a protoplast present. Right? If this particular cell is present in a solution, a sugar solution, this is a concentrated sugar solution. Right? If a plant cell is placed in a concentrated sugar solution, in this case what will happen? Just have a look. Because the outside medium is concentrated, in that the water is less, whereas in case of the plant cell, water is high. Here water high means to say its concentration is high as compared to the outside medium. And we always know Water always moves from a region of a higher concentration to a region of the lower concentration. In that respect, what will happen? There will be a movement of water from inside to outside of the cell. Right? So what has to happen? The cell wall, or you can say the whole cell, has to shrink. Now listen very carefully, because in this case the cell wall is present. It will not allow the shrinkage, or in fact what will happen? Just have a look. All the protoplast that is present, that will shrink. So the protoplast will shrink. Or can I say, because of the movement of water from inside to outside, cell wall remain intact in position. Intact. Cell wall is present in the position only. This particular condition of a plant cell is considered as a plasmolysed state. Plasmolysed state. Now question arises, what is a plasmolysis? Let's have a look. Plasmolysis is movement of water outside of plant cell when placed in concentrated solution. Concentrated solution, that means a hypertonic solution. This is called as a plasmolysis, and the state which arises, this is called as a plasmolysed state, right? So if a question comes, what is a plasmolysis, you have to define that it is a movement of water outside of the plant cell when placed in concentrated solution, right? This is a plasmolysis. This is just how to explain you, this is just a demonstration that how I can explain you to give you a better idea of a plasmolysis. So let's move on to the next question, question number 4. Again the next question, question number 4, 5 and the 6, they are from separate category. So just have a look. What are lysosomes termed as a suicide bags of a cell? Because it is a 2 marks question, so you have to explain in a bit of detail. So just have a look. Lysosome are what? Single membrane cell organelle, right? Right? Which is acidic in nature, and contain various enzymes. That we know about the lysosome. But question arises, why they are considered as a suicide bags? Suppose there are conditions, I'll explain 1 or 2 conditions, right? Suppose on the entry of pathogen means any kind of bacteria or a virus, if they enter into cell, what happens is the enzyme which is present in the lysosomes, they are released. On the entry of pathogen, lysosome attack and kill pathogen. Right. Another activity is that suppose if a malformation is there, a malfunction is there within the cell, or if cell is damaged, right, in that condition lysosome releases enzymes lysosome, they will release all the enzyme which will digest the whole cell, This is because of the presence of hydrolytic enzyme in them. That is why they are termed as, they are called as what? Suicidal bags. Suicidal bags. So, what is a lysosome? Why they are considered as a suicidal bag? These are the single membrane bound organelle which is acidic in nature and contain various enzymes. These enzymes, they are collectively called as a lysozymes. On the entry of pathogen, lysosome attack and kill those pathogen because of the presence of the enzyme like hydroxylase, etc. If the cell is damaged, lysosome releases enzyme which will digest the whole cell, including the whole cell organelles. That is why they are termed as a suicidal bag. In one more condition, suppose some of the organelles, they are not working. In that case also, lysosomes, they releases those enzymes, they will digest that particular organelle so that it can further be replaced. So that is why, that is the reason they are also termed as a suicidal bag. So this is what you have to answer in this question. So let's move on to the next question, question number 5. Name any cell organelle which is a non-membranous. We have discussed about the large number of the cell organelles which are covered with a membrane. But there is one organelle which is not covered with the membrane, and that we have explained also, that is a ribosomes. Non-membranous cell organelle is ribosome. And where these ribosomes are synthesized? Again, I am asking the same question, that they are synthesized at the nucleolus, which is present in the nucleus. Now, to talk about the membranous cell organelles, in this we have an example of an endoplasmic reticulum, ER, endoplasmic reticulum. We have another example like a Golgi. You can take an example of a lysosome. We have n number of example in this membranous bound. You can take an example of a mitochondria. In case of the plant cell, we have chloroplast, nucleus. All of these structures, they are bound by the membrane, whereas the ribosomes are those which doesn't have any as such membrane present. So this was the question number 5. Let's move on to the next question, question number 6. This is again of 2 marks. So the question states, why do plant cells possess large sized vacuole? Yes, you all know regarding the structure of a plant cell. Right. In the plant cell, you must have seen this is a cell wall present. Outside we have a cell wall, then inside you have a cell membrane. Right. In majority of the cases, what we have is a large size vacuole present. So this is what a vacuole is, and vacuole always push the nucleus to one of the side. They push the nucleus to one of the side. Here fluid is filled which is having a majority of the cell organelles as well, which is termed as a cytoplasm. Now, they are asking why this plant cell, this is a structure of a plant cell, Why these plant cells, they have a large size vacuole? The reason is that these vacuoles, they act as a storage of food, they act as a storage of water. Even whatever the excretory products which are formed, they are also stored in the vacuoles, so that overall contamination can be reduced. So let's write about this. Vacuole act as storage of food, water, minerals, right, and also stores excretory products of metabolic activities occurring in cell. Metabolic activities occurring in cell. Now, whatever these metabolic activities which are occurring, they produce a certain excretory product, and which they remain in the vacuoles only, so that cell can be prevented from contamination. Can be prevented from the contamination, right? That is why the vacuole is present only in case of the plant cell. Animal cell, they keeps on moving from one place to another, they don't need to store food as such. That is why they do not have a vacuoles present. So this was the question number 6. This is how you have to attempt 3 to 4 lines. That is enough, no need to write detail, because this is just a 2-mark question. Let's move on to the next question. Question number, again this is a question number 7, a very easy and a very direct question. Now, they are asking the difference between a prokaryotic cell and a eukaryotic cell. So let's write the difference, very easy and direct as well. Prokaryotic cell, here when you are attempting such type of question, one thing should be clear in your mind, that you have to attempt like this. On one side you have to write the one which you have to explain, on the second you should write the second which you have to explain. Don't write in a paragraph manner, rather than point wise is much more clear to examiner, because when we are examiner, when you used to teach, when you used to check the copies, we always look for like within a short duration of time, we have to check like n number of copies. We don't have much of time, we cannot read all these stories. So what you have to do, you have to be precise, you have to write up in a point wise manner and in a comparison way, so that this can be easily visible to the examiner as well. And wherever you see any point important is there, do underline it with the black pen or the pencil, right? So, to talk about the prokaryotic cell, the size is small. I am writing only this thing, that small sized, right? Hardly 10 mm, right? 1 to 10 mm. Right? Whereas a eukaryotic cell is a large size, and the size is around 5 to 100 millimeter. Right? Now, in case of the prokaryotic cell, no as such defined nucleus is present. So second point, no nucleus. Hope you remember this session when I have discussed about the eukaryotic cell. In this case, as no nucleus is present, but in this case, the nucleoid is present. Right? Whereas in case of the eukaryotic cell, the well-defined nucleus is present. well defined nucleus is present. Even in this case, the nucleolus is present. Nucleolus present. Whereas in this case, the nucleolus is also absent. This is third point, right? Fourth point is that in case of a prokaryotic cell, no as such cell organelles is present. No, or you can write no membrane bound cell organelles. No. Whereas in this case, the membrane bound organelles are present. Such as endoplasmic reticulum, Golgi, lysosome, everything is present. Now, in this case, no division of labor is seen. Everything, whether it is related to digestion, respiration, everything is done by the cell only. Whereas in this case, they have a separate organelle for the different function. For digestion, separate organelles is there. For the respiration, separate organelles are there. Or you can write that in this case, no division of labor. Right? In this case, a division of labor is seen. Right? So, to talk about the more difference in this case, here the division, I mean to say the reproduction in this, the reproduction process involved the whole cell. So, the sixth point is reproduction done by whole cell. Now, whereas in case of the eukaryotic cells, we know there are two types of cells. The first one is called as a germ cell and second one is called as a somatic cells. In this case, the germ cells are those cells which are which are helpful in the process of reproduction. So, germ cell perform the process of reproduction. So, as it is a 3 marks question, what you have to do, you have to write a minimum of these 6 differences. If you know more, you can write that as well. So, in case of the prokaryotic cells are comparatively small as compared to a eukaryotic cells. In prokaryotic cells, no nucleus is present, no nucleolus is present. Whereas in case of the eukaryotic cells, nucleus as well as nucleolus both are present. No as such membrane bound organelles is present, whereas in this case the membrane bound organelles like endoplasmic reticulum, for example the lysosome, the Golgi, everything is present in this case. That is why we can say that in prokaryotic cell no as such division of labor is seen. Every work is done by the whole cell only, whereas in eukaryotic cell division of labor is seen. In case of the prokaryotic cell, reproduction is done by the whole cell, whereas in this case the germ cells, the two type of cells we have, the germ cells and the somatic cells. Germ cells, they perform the function of the reproduction, right? So, this is the question number 7, a 3 marks question. This is how you have to attempt, right? Note down this, I will be back. So, students, hope you have noted down the difference between these two. Let's move on to the next question, that is a question number 7. So, the question state that differentiate between unicellular and multicellular organism. You all know about the unicellular organism and the multicellular organism, but here the question is for 3 marks and you have to attempt accordingly. So, on one side write down unicellular organism. On the second side, write down the multicellular organism. Unicellular. Unicellular means the body is made up of only a single cell. Body made up of single cell, right? Whereas multicellular means the body is made up of more than one cell. One cell, right? Another difference is that in case of the unicellular organism, all the body function is done by a single cell only. Whereas in multicellular organisms, a different group of cells, they perform a similar function. That is why they are clubbed under which we call it as a tissue. So, body cell perform, each body cell perform different function. Right? In case of the unicellular organism, the lifespan is short. Whereas in this case, the lifespan is long. Right? They are comparatively longer. Now, in this case, the division of labor is not seen. Whereas in this case, the division of labor is seen. More also you can write, like in this case, the reproduction is performed by the whole cell. Cell divide, they show the process of the binary fission, and this is how they reproduce. In case of the multicellular organism, the special type of cells they are present, which we have discussed also, that the germ cells are present, which perform the function of a reproduction. This is how you have to write the differences between a unicellular and multicellular organism. This was question number 8. Let's move on to the next category. Yes, this is a 5-mark question. Again, a very direct question from the notes which I have already given you. The questions which I have included in the today's session, they are very, very, very important, right? So, draw a well-labeled diagram of a plant cell. Very easy, very direct as well. I'll just give you a rough diagram of a plant cell. This is how a plant cell look like. Right? They are connected. This is called as a middle lamella. Side by side I am labeling. This is termed as a cell wall, whereas this is termed as a plasma membrane. Plasma membrane. Right. We have discussed so many times that in this case, vacuole is present, which act as a storage of food, which act as a storage of the excretory structures as well. So, this is termed as a vacuole. Right. In the vacuole, here the sap is filled. So, I am writing over here cell sap. The covering is termed as a tonoplast. Right. Remember one very, very, very important structure. In this case, a well-defined nucleus is present, which are having a nucleus pore present. In this case, a nucleolus is also present, and these are the chromatin fiber present. So, this is termed as a nucleus. This is termed as a nucleus pore. This is termed as a nucleolus. right? Now, another important thing is that in this case, such type of structures, they are present. Membrane structures, which is termed as a endoplasmic reticulum. Sometimes these endoplasmic reticulum, they are associated with the ribosomes. That is why they are termed as a rough endoplasmic reticulum. The dot which I am drawing right now, they are ribosome. So these are called as a rough endoplasmic reticulum. Likewise, we have some Golgi bodies also present, right? Golgi. Some of the ribosomes, they remain free like this. So these are termed as a free ribosome. Free. They are free with plant cell, right? Because they are present free in the cytoplasm. Very important structure. In this case, a chloroplast is also present. These are the granums which I am drawing right now. Hope you are getting what I am trying to say. This is a granum termed as a chloroplast. Now, as we have discussed so many structures, how can we forget about the powerhouse of a cell? They do need energy as well. So, let's draw the powerhouse of a cell. This is termed as a mitochondria. Along with that, so many other structures, they are also present. And one more thing, how can we forget about the cell shape, which is maintained by large number of the microfilaments and the microtubules. Microfilaments, and how can we forget about the microtubules, right? This is what you have to draw. Along with the cytoplasm also you can label. So this is how you have to draw. If such type of questions comes in a 5 marks, draw a well labeled diagram, neat and clean diagram. You can, if like your teacher permits, you can use different colors also and try to do the majority of labeling on one side. Like if you wish so, that looks good. In fact, if you do not have space both sides, that is also fine. Each and every separate organelle that you should show, though I was not having as much of time, but we have discussed in the session. You know about the chloroplast, that is also covered by 2 membranes. You can show the 2 membranes are also there. Whereas in case of the chloroplast, you can also say that in this case a small DNA is also present. They do have their own genome. Likewise, you can draw in case of the mitochondria as well. The smooth endoplasmic reticulum I forgot to draw, you should draw that as well. This is how you have to attempt this question. Let's move on to the next question, that is a question number 10. Read this question. By the time, let me prepare this board for you. What are genes and what is the difference between the genes and the chromosome? So, if I have to explain the genes, what are genes? Genes are a stretch of DNA. Let's write. Genes are stretch of DNA. What is the full form of DNA? That is a deoxyribonucleic acid. Deoxyribonucleic acid. Genes are stretch of DNA which also act as heredity material, which also act as a hereditary material. Once again, understand what I am trying to say. The genes are the stretch of the DNA. Stretch of the DNA is there and they code for a specific protein. And you can say a functional unit. Functional unit which code for specific protein. Specific protein, right? Now, what are these chromosomes? Chromosomes are a thread-like structure which not only consist of this DNA, rather than in this case the proteins, they are also present. They are very beautifully wrapped around a protein, right? We have discussed also regarding the histone protein. This is what you have to explain in this case, right? So, what are chromosomes? Are thread-like structure in which the DNA is there and DNA is associated with the protein as well. What is the nature of DNA? Is it positive charge or negative charge? This is negative charge, because as you can see, this is a deoxyribonucleic acid. In this case, the phosphate is present. So, overall, to neutralize this charge, the proteins which is present in them, that is called as a histone. Very beautifully wrapped, which we call it as a nucleosomes. Why is the inner membrane of the mitochondria folded? The reason is that to increase the surface area. The first, to increase the surface area. Why we need to increase the surface area? Because majority of these reactions by which the ATP is synthesized, they occur on the inner mitochondrial membrane. Because this inner mitochondrial membrane has F1 particle. Now, what are these F1 particles? They are also called as an oxysome, in which one enzyme is present, that is the ATP synthase, which help in the synthesis of ATP. And we know that the energy currency of a cell is ATP only. This is how they are synthesized, and they are synthesized within the innermost membrane where these particles are present and they are highly folded. Now, these folded structures which are present, they are also called as a cristae. They are also termed as a cristae. Draw a well diagram of mitochondria in this, right? Do explain why these folds, they are important. And in the first, if you wish so, you can draw the diagram also. Because it is a 5 marks question, so you have to attempt in a bit of a long answer type question type. And wheresoever the diagram, you know, you must draw the diagram. Diagram should be neat and clean. And one thing that should be clear in your mind, never ever draw the diagram with a pen. Always draw a diagram with a pencil. Even in your notebook, when you try to write whatever I have written on the board, if you try to make your notes as well, in that case also you should use pencil. No need to use pens, right? So, this is all about today's session. We have discussed the 10 questions separate each from the different category. Hope everything is clear to you, not all clear to you. So, we will meet you in next session. If you face any problem in this chapter, you can directly ask us. So, thank you so much students for watching this.